Hello, everybody, and welcome to our webinar today entitled Alzheimer's Disease Drug Development and the Changing Alzheimer's Disease Therapeutic Landscape. My name is Shannon Woodruff, and I'm the Marketing Manager here at Clinilabs. I'll be your moderator today. It's my honor to introduce our guest speaker, Dr. Jeffrey Cummings. Today's webinar is being presented by Clinilabs Drug Development Corporation. Before I introduce today's speaker, I'd like to cover a few housekeeping items. Today's webinar is being recorded. We'll be able to share a link with you after the event is over, and we welcome you to revisit the content and share it with your colleagues and people you might work with. We also invite your comments and questions. Please identify the interaction panel on the right-hand side of your screen. Please note that there's a questions tab and a chat tab. So if at any point during the presentation you have a question, please add it there and we'll hold it for the Q&A portion at the end of the event. Our presenter today is Dr. Jeffrey Cummings. Dr. Cummings holds many positions, including the Joy Chambers Grundy Professor of Brain Science, Director of Chambers Grundy Center for Transformative Neuroscience, and among others. Dr. Cummings is globally recognized for his contributions to Alzheimer's research, drug development, and clinical trials. He's been recognized for his research and leadership contributions in the field of Alzheimer's disease through numerous awards and achievements. He's authored or edited over 43 and published over 800 peer-reviewed papers. Without further ado, I'd like to introduce our guest speaker, Dr. Jeffrey Cummings. Take it away.
Great. Thank you, Shannon. Thank you for that very nice introduction. Welcome, everyone. Really great to be on this platform and sharing these ideas with you. So I will be talking about drug development and the changing Alzheimer's disease therapeutic landscape. It's a fantastic time in Alzheimer's disease research, and you are all part of it because you're doing clinical trials, and clinical trials are the way forward to discovering new therapies, of course. So it just couldn't be better. Here are my disclosures. I've consulted for a lot of different biotech companies and pharmaceutical companies to try to find the best possible therapies for Alzheimer's disease. Of course, I will be talking about unapproved medications in this lecture. That's really the point of this lecture. Here are the things I'd like to cover in the next 30 or 35 minutes. Talk about an overview of drug development, just so we're using all the same terminology. I'll talk about our work with the pipeline. We do an annual pipeline publication that is followed by many, and I'll show you how that works. I'll talk about the target categories for Alzheimer's disease drug development and how we derive those categories. I'm going to emphasize monoclonal antibodies because they are so much in the news right now. You are all probably doing trials with monoclonal antibodies, and we're going to have a lot of news about monoclonal antibodies soon. I will talk about the role of biomarkers in clinical trials. They have been truly transformative in our thinking about clinical trials and our ability to do clinical trials well. And I'll finish with some trial observations, some facts that we observed as we were doing our pipeline review. So let's start with an overview of drug development. I think most of you know how this works, but just so that we're all using the same terminology. Of course, it begins with non-clinical discovery and animal testing. So a drug starts as a white powder in a laboratory usually. Is tested in a genetically engineered animal to see whether it affects the target mechanisms. Now, animals are poor predictors of success in humans, but you wouldn't advance a drug to humans that didn't work in the mice. So there's a kind of a gateway function for the role of mice in drug development. And of course, you must know the safety. You all know that. You're constantly monitoring the safety of your patients in clinical trials. So that begins with the animal work. We then progress to phase 1, where we start in the first in human. These are truly heroic individuals who are willing to take the very first exposure to a drug that has only been studied in animals until then. So first, single ascending doses are studied. So a person takes one dose, and you see how they do, or a cohort of individuals. Then you usually double that dose with another cohort and see how they do. And you're just giving each of these cohorts one dose, single ascending dose, or SAD. If that all goes well, then you want to go back and test multiple ascending doses because, of course, you're going to give multiple doses over time. So you do the multiple ascending dose study, the MAD study, in which, again, you expose someone usually, or a cohort of individuals, usually for 28 days. Sometimes it's shorter, as short as 14 days. Then you would double the dose usually and go to the next cohort and double the dose until you found either the maximum tolerated dose or the maximum feasible dose. And that determines the doses that are advanced to phase 2. So phase 2, for me, most exciting of all of the phases, it's proof of concept and dose finding. You have 400 to 800 actual Alzheimer's disease patients, and now you're trying to determine, is the drug doing what we think it should do in order to modify the disease or to control symptoms in some way? If that all goes well, then it's advanced to phase 3. These are the confirmatory trials where clinical benefit must be shown, except in the case of accelerated approval, where approval on the basis of a biomarker with a commitment to a confirmatory clinical trial is the way forward. After that, of course, the FDA then reviews, and they would be responsible for deciding about accelerated approval. So there would be a comprehensive review of both the efficacy and the safety to decide the next step for that drug. Now, I'd mentioned that I'd like to emphasize biomarkers. So here's the way that biomarkers fit in to this development program. In the animals, there would be measurable biological changes first in assays that are not in animals, and then in the animals themselves. Then there would be safety biomarkers that are collected in phase 1, like EKG and liver functions, and we've used those for so long we don't even think of them as biomarkers, but of course they are. Then you go on to proof of concept, where you would have a biomarker for the diagnosis of the patient to make sure that they have Alzheimer's disease. You would have a biomarker for target engagement. Is the drug engaging the target that we want it to in the human being so that it could possibly have a therapeutic effect? If possible, you do some dose response finding with the biomarker, and of course you have safety biomarkers. In phase 3, again, you would use biomarkers for the diagnosis of the population. You would then collect data if it's the trial for a disease-modifying compound. You would then collect biomarkers which either favor or fail to favor the presence of disease modification by the drug. And again, you would be collecting safety biomarkers. As the drug goes to the FDA for review, the biomarkers would be included in the labeling in the clinical pharmacology section. The biomarkers are not an indication, that the clinical diagnosis is the indication. And as I mentioned, occasionally the FDA would make a decision to use accelerated approval, as they did with aducanumab. So biomarkers have a very important role all the way through the drug development process. And imagine that only a few years ago, we did not have such biomarkers. So we've been able to use biomarkers to change a shot in the dark to a shot on goal. As we think about drug development, we want to think about, well, what constitutes a quality program for drug development? So some years ago, we wrote a paper called The Rights of Drug Development. So what are the rights? Well, the first one is the right target. Is the target druggable? Is it supported by genetics? is a critical mechanism, so that if you turned it up or turned it down, would it have an effect on the disease process and the clinical course of that patient? Then, of course, the right drug. Does your white substance have drug-like properties? How about the absorption, distribution, metabolism, excretion, and toxicity? What do we know about that? Is a maximum tolerated dose established? That is the way you decide what the highest dose could possibly be. And of course, when you have a negative outcome on a trial, you want to make sure that you have tested the appropriate dose before you give up on that drug. You need to have the right biomarker. I've been emphasizing that. So patient selection, target engagement, disease-modifying outcome, safety are all the right biomarkers. What about the right participants? Well, this is a new part of our discussion now. We know that Alzheimer's disease has a long preclinical phase, about 15 years before symptoms appear. Then there's about a 5-year stage of mild cognitive impairment before dementia appears. And then there's about a 10-year period of progression through mild, moderate, and severe Alzheimer's dementia. So what is the right population for the drug that we are developing? That's a critical question. And for the most part, we think that drugs will do better if we place them earlier in the process before there has been a lot of damage to the brain. On the other hand, the cholinesterase inhibitors, the drugs that we have available to us now, are approved for mild, moderate, and severe Alzheimer's dementia. So we know that it is possible to push the brain, even though it is heavily involved in those late stages. And then finally, what about the right trial? And this is what your teams are so important for. Is it the right trial? Is it well conducted? Can we depend on the data? Is the data submitted in a reliable way? Are the protocol violations avoided? These are all the things that constitute a great trial, and they're terribly important for a successful drug development program. And of course, the design of the trial is very important. Is it large enough to be able to draw a conclusion? Have all the right doses been tested? Are the right outcomes used? So the right trial is the final of the 5 rights. And as you look at development programs, you can sort of have a checklist in your mind about, well, do they have the 5 rights? Surprisingly often, drugs are advanced where we don't know some critical aspect of the drug. Tell you a little bit about the background of our annual review, where we look at all of the drugs in the pipeline. We started publishing this in 2016, and we have published it every year since. It's based on the information in clinicaltrials.gov. And you know that every clinical trial is required by law to be registered on clinicaltrials.gov if that trial is to be conducted in the United States. Many trials that are conducted ex-US are also registered on clinicaltrials.gov, but not all of them. So it is exhaustive with regard to US trials, and it is comprehensive with regard to global trials, but there are some trials that are not registered. Each year we have an index date. So whatever we say in our article was true on that day. So here, this year, the index date was January 25th, 2022. So we presented all the drugs in clinical trials on that date. So if a drug entered the clinical trial on 1/26, one day later, it would not be included in this review. And of course, you have to have a cutoff time so that you can prepare the article and then publish it. But so it's important to know how the index date works. We use artificial intelligence and machine learning to interrogate the database. We also use the Common Alzheimer's Disease Research Ontology, or CADRO, for the mechanistic classification. So I'm going to talk about the mechanisms of drugs, and the way we approach that is CADRO is a list of processes identified in Alzheimer's disease, and they represent the targets for drugs that might modify the course of Alzheimer's disease or improve the symptoms of the disease. So we use the CADRO system to allow us to think comprehensively about the mechanisms that drugs address. And this is all funded through the NIA and something called the Alzheimer Clinical Trial Innovation or Action Initiative. Here's the main figure from our annual publication. And this figure is widely shown in lectures, and we're very proud of it. So the central circle are all the phase 3 drugs. So these are the drugs that are furthest along in development and could be presented to the FDA if they succeed at phase 3. The middle circle is phase 2. So these are all the compounds that are currently in that proof of concept and dose finding stage of drug development. And the outer circle is phase 1, those drugs that have just come out of the animals and are now going to be tested in the first in-human trials. Now, we know that many of the phase 1 trials are done ex-US and not registered on clinicaltrials.gov, so we would not be able to find those drugs, and that's part of the reason that it looks like there are very few phase 1 drugs. There are actually more than this. Now, the green slice of the pie at the top, those are all biologicals, so mostly monoclonal antibodies, although there are some other biological compounds as well. The purple slice of the pie on the right are disease-modifying small molecules. So these are largely drugs that you would take by mouth, and they are intending to slow the progression of Alzheimer's disease by intervening in some critical process. The blue slice of the pie on the left are drugs that are being developed for the neuropsychiatric symptoms of Alzheimer's disease. For example, agitation is a particularly hot area right now in terms of drug development. And then the orange slice of the pie on the left are cognitive enhancing agents. So the currently approved drugs such as cholinesterase inhibitors and memantine are cognitive enhancing drugs. They aim to improve cognition, but they do not target the slowing of the decline in the course of the illness. And in general, we're going to be using both types of drugs together. Each of the colors represents the CADRO category for that drug, and each of the shapes represents the population that is represented in that trial. Well, altogether, there are 143 trials for there are 143 agents in 172 trials. So there are always more trials than agents because some agents are in more than one trial. The drug might even be in a phase 2 proof of concept trial, but still in a phase 1 drug-drug interaction trial. So you see some drugs represented in multiple trials. In phase 3, there are 31 agents. 21 of these are disease-modifying molecules, so most of the emphasis in the pipeline is on disease modification rather than on cognitive enhancement or neuropsychiatric symptoms. If we look at phase 2, there are 82 agents there. Again, 71 of the 82 are disease-modifying agents. So that is the real emphasis in the pipeline. And as we look forward to drugs coming into your trials, they're largely going to be disease-modifying drugs. And as we think about those drugs eventually, hopefully, making it into clinic, then they will largely be disease-modifying drugs. Phase 1 has 30 agents. So altogether, disease-modifying therapies represent 83% of the agents in the pipeline. About 10% are cognitive enhancers and about 7% are for neuropsychiatric symptoms. Of great interest to me is that 37% of the drugs in the pipeline are repurposed. That means that they're approved for some other indication, but there are good reasons to believe that they should be tested in Alzheimer's disease because of some shared mechanism between the approved indication and Alzheimer's disease. So, for example, a drug might be approved in cancer, but it has some properties that make us think that it would be useful in Alzheimer's disease, or it might be approved in Parkinson's disease, and we would think it would be useful in Alzheimer's disease. So those are repurposed agents. Along the left of this graph, I show you the CADRO categories. So beginning at the bottom, amyloid, tau, inflammation, synaptic function, metabolism and bioenergetics, protein metabolism, vasculature, oxidative stress, epigenetics, neurogenesis, and transmitters, and others. So the blue color is phase 1, the green color is phase 2, and the red is phase 3. So what you can see immediately is how different aspects of the cadro classification, that is, different disease processes, are being addressed with different levels of enthusiasm in the pipeline. So the mechanism with the greatest number of compounds in trials is inflammation. That reflects the great growth of our understanding of inflammation in Alzheimer's disease in recent years. You see that synaptic plasticity is also well represented in the pipeline. Tau and amyloid have drugs in the pipeline, and then a smaller number of drugs for the other categories, but still a very important set of drugs that are being tested, for example, in metabolism and bioenergetics, where we're testing many of the drugs that have been approved for diabetes. And diabetes is both a risk factor for Alzheimer's disease and shares some pathophysiology with Alzheimer's disease. And those drugs are in the metabolism and bioenergetics category. One point I'd like to make is that amyloid represents 14% of the pipeline and tau represents 9% of the pipeline. So altogether, that represents 23% of the pipeline. I'm sorry, 27% of the pipeline. I see there's an error there. And 73% of the pipeline are non-canonical targets. We often hear, well, we're devoting too much time, we're devoting too much energy, we're devoting too much money to amyloid. But you see, actually, it's only 14% of the pipeline and tau 9% of the pipeline. So other targets are actually much more represented in the pipeline than amyloid and tau. I want to go through a few of these cadro areas in more detail because I think you'll find some of these observations interesting. So here I'm showing you the inflammatory pathways on the left, and you can see at the top a resting microglial cell being transformed into an activated microglial cell. That has good aspects and bad aspects. So the good aspect is that activated microglia can ingest amyloid, can ingest tau, and can benefit brain function. However, those cells also stimulate a pro-inflammatory set of cytokines that are toxic to nerve cells and therefore increase the toxicity to the brain. And we're looking for this balance between the function of microglia that we need without stimulating the pro-inflammatory chemicals that can be released in this process. And you see that this activation can be done by amyloid. It can be done by neurodegeneration and cell death, that inflammatory cells can enter from the periphery, and that this is a final common pathway for many aspects of Alzheimer's disease. Now, what interests me is you look on the right and you see a box with all of the drugs that are currently in the pipeline that have inflammation as their primary target. And what you see is that almost none of the drugs have the same target within the inflammatory pathways. So you see, I'm not going to go through all of these, but you see that at the top, there's a TREM2 antibody. So TREM2 is one key aspect of Alzheimer's disease inflammation. You see that there's a non-steroidal anti-inflammatory drug. You see that there are regulatory T cells that are being tested. You see that there are cytokine and leukotriene antagonists that are being tested. So we're going to learn a ton about inflammation in the near future as these trials read out and we begin to understand which of the pathways seem to be more important in terms of Alzheimer's disease. And we will also see whether there are logical combinations of drugs that might address inflammation and therefore work synergistically to amplify the anti-inflammatory impact of the treatment. Now, in a similar way, I want to take you through the drugs that are impacting synaptic integrity and synaptic plasticity. So again, on the left, you see a model of this, and you see the synapses at the top. These are adversely affected by many of the processes that are part of Alzheimer's disease. Here you see the toxicity of the A beta oligomer. You also see the toxicity of the neuronal death, as well as, and not shown here, the toxicity of inflammation. Now, important to me is that synapses are organized into circuits, and therefore it is circuit disruption which ultimately is responsible for the neuropsychiatric and cognitive symptoms of Alzheimer's disease. And the fact that these are organized into circuits also gives us new biomarkers. For example, EEG is a biomarker for circuit function, and a functional MRI is a biomarker for circuit function. So we have biomarkers for the synapse, measurable primarily in the spinal fluid, and we have biomarkers from the circuit, such as the ones I just mentioned. Again, look at that list of drugs on the right and note that nearly each of them has a unique target on the synapse. So you see that there are GABA B modulators. You see that there's a PKC inhibitor, that there's a gingipane inhibitors, that there are neurotropic agents and sigma-2 antagonists. So you see that there's a plethora of drugs where the idea is to try to influence synaptic function. And again, by looking across the landscape of synaptic agents, we're going to be able to become more informed about the pharmacology of the synapse as it is modifiable by anti-Alzheimer's disease therapeutics. I mentioned these anti-diabetic agents that are being assessed in clinical trials. In phase 3, for example, well, let's start with the figure on the right now. So in diabetes and hyperglycemia, you have increased insulin, you have increased glucose, you have fat cells, and all of these activate the advanced glycation end products in the brain. Similarly, I've noted that peripheral inflammation can lead to central inflammation and reactive oxygenation species or oxidative injury. All of these processes then contribute to increased A beta, increased neurofibrillary tangles, and increased insulin resistance, all of which are mediators of neurodegeneration. So you can see how the biology of Alzheimer's disease and obesity is intimately related to the biology of Alzheimer's disease. And therefore, it makes sense that we would be testing some of the anti-diabetic agents in terms of the treatment of Alzheimer's disease. So in phase 3, we have metformin, an insulin sensitizer, and semaglutide, a GLP-1 agonist. Some of you may be involved in this trial. The GLP-1 agonists are interesting because they are insulin sensitizers, but they are also profoundly anti-inflammatory, and that's thought to be the main action of this in Alzheimer's disease. And we know that diabetic patients who took the GLP-1 agonist are less likely to develop Alzheimer's disease than other diabetics. And diabetes is a risk factor for Alzheimer's disease. So this all points to good reasons to be testing these drugs in humans. In phase 2, we have an SGLT2 inhibitor, we have a PPAR agonist, and we have insulin itself. So we're testing a lot of different anti-diabetic agents in this pipeline. What about vascular factors? We all worry about stroke and its relationship to Alzheimer's disease. And I want to make one major point in looking at this particular part of the pipeline, which is that this is the aspect of the pipeline where we see current combination therapies. I think most of us are convinced that Alzheimer's disease is so complicated that the likelihood that one drug is going to be able to do enough to achieve all we want for our patients is very unlikely. And we're going to need combinations of drugs to keep our patients functioning at the highest level for the longest period of time. So you see that there's a phase 3 trial of a combination of losartan, amlodipine, and atorvastatin. These drugs are usually used for hypertension and cholesterol control, but they are also thought to be active in Alzheimer's disease. You see that there's a phase 2 trial for a combination of telmisartan and perindopril. So again, you see that these combinations are being tested in the vascular part of the pipeline in ways that we do not see in other parts of the pipeline. they're going to be highly instructive. Now let's go to tau-directed therapeutics. I'm emphasizing here both small molecules and monoclonal antibodies. So you see the biology of tau, beginning with transcription and then translation, organization into the microtubule, but then as it comes apart in the tauopathies or the tauopathy portion of Alzheimer's disease, the tau comes off of the microtubule, forms dimers, trimers, oligomers, protofibrils, and filaments, eventually forming the neurofibrillary tangle of Alzheimer's disease. Now, very interesting, we've learned in recent years that there is cell-to-cell propagation of the tau molecules. So that is how Alzheimer's disease spreads in the brain from a tau perspective. So the tau you see in the lower left is in the presynaptic region. It is transferred then through the synapse into the postsynaptic neuron, where it then kills that neuron, but is also released into the next neuron. So you see that it begins by very little tau in the brain with the Braak stage 1 and 2, progressing to more, it's Braak stage 3 and 4, and finally the worst of Braak stage 5 and 6. Now, the drugs that we're looking at to try to inhibit this process include aggregation inhibitors, 2 of those. There's one vaccine. There are several monoclonal antibodies that are being tested or have recently been tested. There's an antisense oligonucleotide that is administered intrathecally to interfere with tau biology. There's a GNA case inhibitor from Lilly that is being tested, and a microtubule polymerization agent, nicotinamide, whose target in this trial is tau. I'm going to move on now to amyloid. I'm going to start with the small molecules. And you see there on the left the biology of amyloid. So it starts off as the amyloid precursor protein, which is a huge protein, 700 amino acids long. It is then cleaved by beta secretase, followed by gamma secretase to free up the A beta monomer that is 42 amino acids long. And that tends to aggregate into the oligomer. There's a lot of evidence that the oligomer is quite toxic to nerve cells. And then the oligomer goes on to fibrilize and eventually to form A beta plaques. So we can intervene in this process in a multitude of different places. So one way is, as you see in the table on the right, you can enhance the non-A beta, so-called alpha secretase pathway. So it doesn't go through beta and gamma secretase, and therefore A beta is not generated. There are 2 drugs that do that. You could reduce APP by decreasing RNA transcription. There's one drug that does that. You can decrease pyroglutamate A beta, and there's a drug that's being tested for that, or activate an ABCA1 amyloid transporter, or inhibit A beta aggregation. So there are multiple ways that are being tried to interfere with the A beta pathways. Now, the monoclonals, of course, have had great attention this year, a lot of controversy, and I'll go through that a little bit. Again, you see our targets on the left, and then you see the monoclonals on the right. So aducanumab has been approved. Its targets are A beta plaques and oligomeric A beta. Donanemab is in several trials. It targets the A beta plaques, particularly the pyroglutamate form of A beta. It is in phase 3. Lecanemab targets the A beta plaque and protofibrillar A beta. It is also in phase 3. Gantenerumab targets A beta plaques and oligomeric A beta. It is in phase 3. And solanezumab is in a phase 3 trial, a prevention trial. Of course, solanezumab has failed in treatment trials of patients who have symptomatic disease, but it is being tested in patients who began with no symptoms, although they had amyloid in the brain. And that trial will read out in 2023. Now, the excitement here is that both lecanemab and gantenerumab will read out before the end of the year. So we're going to have information very soon about these 2 drugs, and we will begin to understand the relationship, if any, between plaque lowering and slowing of cognitive decline. So this just couldn't be a more exciting time than it is for Alzheimer's therapeutics. There are some novel developments in the pipeline, amyloid vaccines, tau vaccines, antisense oligonucleotides, I mentioned that. Epigenetic interventions and stem cells are being looked at, and also in phase 1, we see epigenetic interventions and stem cells. So the stem cells, they're of great interest, particularly to the public. They've really caught on to this, but they are not very advanced in the pipeline. They're very difficult to use and control, and so that will be, I think, a slowly developing portion of the pipeline. I want to talk a little bit about aducanumab because it is the only approved disease-modifying agent in the United States at this point. Of course, it was based on, the approval was based on the amyloid reduction. So in the figure on the right, in the upper two figures, the upper panel, you see a patient who was on placebo for a year. And the red color represents the ligand attached to amyloid. So you see that after a year's exposure to placebo, there was no change in the amyloid. Now let's look at the bottom panel. These are patients who received 10 milligrams per kilogram for 12 months. You see on the left, again, there's a lot of red color. That's because the amyloid was required for participation in the trial. And then on the right, you see the very dramatic clearing. That scan is a negative scan. That patient would no longer qualify for a diagnosis of Alzheimer's disease. So these drugs are producing, indeed, when I say these drugs, I mean plaque-directed monoclonal antibodies are having dramatic effects on plaque burden. So the FDA approved this based on the effect on a defining pathophysiological feature of AD, which is A beta removal. There's a lot of controversy about whether this was the right decision, and a follow-up confirmatory trial is required. And as I say, we have 2 drugs coming soon that will bear importantly on this relationship between plaque lowering and clinical slowing. The data in the trial were supportive of that, and it led to, as I say, accelerated approval. Now, just to give you a little bit of information about the 2 trials that were so controversial. In the EMERGE trial, the clinical dementia rating sum of boxes, the CDR-SB, was the primary outcome, and it demonstrated significant slowing. So there was 22% slowing that was statistically significant. The secondary outcomes, and these are nominal p-values or nominal derivatives of the trial. The ADL measure was slowed by 40%, the cognitive measure by 27%, and the mini-mental by 18%. Now, the ENGAGE trial was similarly designed, in fact, identically designed, also had CDR as its primary outcome and was negative. However, when you look at ENGAGE and look at those patients who had 10 milligrams per kilogram for 14 months, which was the target exposure, then those patients had 30% slowing, as that same group had in the EMERGE trial. So there does look like there is a pharmacokinetic relationship, a pharmacodynamic relationship, but we need to confirm that in additional trials. There was plaque reduction, and there was also very interesting downstream effects. And I'm showing you the tau PET scan on the left. And you can see that on the right, and you can see that on the farthest right image, there's a substantial signal from the ligand showing neurofibrillary tangles in the medial temporal region. And then in the next image over, you see that there is a reduction in the neurofibrillary tangle shadow following the exposure to aducanumab. So there was a reduction in tau, both CSF measures of tau and the PET measures of tau. Excuse me. Now, aducanumab and all of the monoclonal antibodies do produce ARIA, amyloid-related imaging abnormalities. It's really a mechanism-based observation. As these drugs pull amyloid out of the brain, out of the plaques, they also pull amyloid out of the blood vessel because there's a lot of deposition of amyloid in the blood vessel. And when that happens, then temporarily, particularly during the titration phase, there's likely to be extravasation of fluid and occasionally extravasation of blood into the brain. Most of it is completely asymptomatic, 75%, but 25% is symptomatic and about 1% is very symptomatic. So we have to keep on top of this during the administration of monoclonal antibodies. So you see here the pathway that we published in terms of how we think this should be done. All symptomatic patients who have ARIA would have treatment interrupted until the ARIA resolved. If the ARIA was asymptomatic, if it was mild ARIA on the MRI, then we would continue dosing. But if it was moderate or severe, and even though asymptomatic, we would suspend treatment until it resolves. We also came up with a series of stopping rules, which you see at the bottom of the figure. Any macrohemorrhage, more than one area of superficial siderosis, more than 10 microhemorrhages since treatment began, more than 2 episodes of ARIA or severe symptoms of ARIA. So we can manage ARIA, but it is an important responsibility that we take on when we administer monoclonal antibodies. Now, I want to talk just a little bit about biomarkers. Here are the 7 categories or context of use that are recognized by the FDA. So a biomarker can be susceptibility and risk biomarker, like the ApoE genotype. It can be a diagnostic biomarker, like amyloid PET. It can be a monitoring biomarker, and again, amyloid PET or tau PET could be monitoring biomarkers. It can be a pharmacodynamic biomarker. Here it would be usually one of the ATN, that is amyloid, tau, and neurodegeneration biomarkers. It can be a predictive biomarker that predicts a response to therapy. For example, the presence of the ApoE4 genotype predicts a greater degree of ARIA in patients receiving monoclonal antibodies. So that's a predictive biomarker. Prognostic biomarkers are those that predict provide a prognosis for the patient. That is, will they decline? And we know that p-tau 181 and p-tau 217, if elevated in the plasma, indicate that that patient is likely to decline. And then there are safety biomarkers. We've mentioned the use of MRI to monitor ARIA, as well as liver functions and EKG. So there are a series of biomarkers that must be qualified for use in a clinical trial. And just to show you how this is being used, here's the donanemab Trailblazer trial. You see in the upper left that a PET scan, a tau PET scan was done. And patients had to have some tau, but not too much tau. So it was the middle level of tau that was desirable for participation in this trial. And in the upper right, you see that as amyloid was decreased in the brain, there was also a reduction in the p-tau 217. So the p-tau 217 plasma measure became valuable as a way of following the amyloid reduction. But of course, you don't want to do multiple amyloid PET scans, so having a plasma measure that is informative about that plaque reduction is extremely valuable. In the lower left, you see tau PET as a pharmacodynamic biomarker in different portions of the brain, showing less accumulation of tau during the course of therapy. And in the lower right, you see the actual amyloid reduction. You see that by 24 weeks or just 6 months, there's already marked reduction of amyloid overall, mean reduction. And you see that that continues through week 76. So biomarkers are again being used throughout the drug development process. This is just a partial set of examples. Now, just to conclude, I want to show a few observations that I was struck by as we went through our pipeline paper. Let's think first of all about, well, what populations are the trials in? Well, only 6% of all the trials are prevention trials that involve cognitively normal individuals. About 40% are in mild cognitive impairment or mild cognitive impairment and mild Alzheimer's disease dementia. So right at that transition point between MCI and Alzheimer's dementia. That's a very popular group of patients, and there's a lot of competition for those patients, as some of you likely know. There are fewer trials, about 26% in mild to moderate, and even fewer, 8% in moderate to severe. Where are the trials done? This is a global enterprise. And you see that particularly at phase 3, 40% of all trials involve both North American and non-North American sites, so involving the whole world in terms of drug development. In phase 2, there's more concentration on North America. 54% of the trials are done only in North America. And in phase 1, there's a pretty good division in North America only or non-North America only, where sponsors go to a phase 1 site in Australia or the UK or somewhere like that. Who sponsors trials? This is very important. You see that in phase 3, 68% are sponsored by the biopharmaceutical companies only, and 11% are public-private partnerships. That would be NIH, for example, plus a pharmaceutical company. So overall, 79% or 80% of trials critically involve the pharmaceutical industry. But if you look at phase 2, you see that academic medical centers and NIH are very involved in phase 2. This is that proof of concept stage. This is where we're really learning about the mechanisms that might impact our patients. So this is a critical part of drug development. And you see that it's about equally represented between academic medical centers and the biopharmaceutical industry. So just to finish off, of course, this is a very complex ecosystem of which you are all a part. It involves academic medical centers, biotech, pharma, capital, advocacy, contract research organizations, sites, regulatory, payers, training, NIH. All of this must work together in order to have a successful drug development program. So if I can just summarize, there are many mechanisms represented in the pipeline. Inflammation and synaptic plasticity stand out as common targets. Combination approaches are uncommon except for the vascular section of the pipeline. Amyloid and tau are common targets, but they comprise a minority of pipeline agents in the pipeline. Biomarkers are enormously useful, and we need a lot more of them. Most drugs in the pipeline are disease-modifying therapies. So those are the drugs that you're going to be seeing in your clinical trials. Aducanumab is the first approved disease-modifying therapy for Alzheimer's disease, but we are right on the verge of having a lot more information about monoclonal antibodies and whether they have an effect on clinical decline. So this is my group that I work with and that support me in all of these studies, and I thank you very much for your attention today. Thank you.
Thank you so much, Dr. Cummings. That was a very interesting presentation. We do have 2 questions that came in. And just a reminder for those watching, if you have any questions, now would be the time to put them in the questions tab. First question for you, Dr. Cummings. Are we close to a therapeutic intervention that can prevent Alzheimer's disease?
Yeah, really great question. Thank you, Sabi, for that. Are we close? I think we are close. So I've mentioned the progress in the monoclonal antibodies. I've mentioned that 6% of trials are currently in cognitively normal people. So when given to a cognitively normal person, if we prevent the emergence of symptoms, we would say, well, that is a prevention intervention. And the monoclonals, look like they might be able to do that. We have several monoclonals in prevention trials right now. Lecanemab is in a prevention trial. Gantenerumab is in a prevention trial. As I mentioned, we're soon going to get a readout on solanezumab in a prevention trial. So we're just learning at an enormously fast pace right now about both prevention, which we really need, and the possible therapies that might work for prevention.
Great. Next question. What are the most promising drugs in the AD drug pipeline?
Most promising drugs. I've emphasized the monoclonals because they are just getting enormous media attention right now, enormous scientific attention. And you do see that they have a dramatic biological effect. We've never seen something like plaque clearing in the way that we're seeing it with the monoclonal antibodies. So we can see that these drugs are really having the biological effect that we think is important. Now, we still have a lot to learn. The amyloid PET scan shows us only the plaque. And there are a lot of other species of amyloid, as I showed you in my diagram, in the brain. And we don't know what these monoclonals are doing for those other species of amyloid. So right now, I regard the monoclonals as the most promising drugs in the pipeline. I haven't emphasized in this talk the drugs addressing neuropsychiatric symptoms in Alzheimer's disease. But since you're asking about the most promising drugs, another drug that had a very successful phase 3 trial recently is brexpiprazole for agitation in Alzheimer's disease. And brexpiprazole had two previous trials that indicated that it was reducing agitation in Alzheimer's. This the third trial, I think, confirms that, and I think it is very likely that the FDA will approve brexpiprazole. Of course, I don't know that, but I think the data look quite good, and that will, if it is approved, that will be the first drug approved for any neuropsychiatric symptom of Alzheimer's disease. So I just love to see that threshold crossed for the first successful neuropsychiatric drug in Alzheimer's disease. So those are drugs that would be on my list as most promising. There are a few others as well, but I won't take time to talk about them right now.
Okay. Looks like we have one more question. We are learning that microbiome relationship to various diseases could change the way we approach treatment of those diseases. Is there a relationship with microbiome and Alzheimer's?
That is just a great question, Michael. Thank you for that. We're just learning so much about the microbiome, aren't we? We see that it's different in Alzheimer's disease than normals, in Parkinson's disease in normals, in multiple sclerosis in normals. And it really looks like it might be contributing. And there are several ways in which it might contribute. One is when you give a drug, of course, it's partially metabolized by the microbiome. So you have that effect. When you give a drug, you might have an effect on the microbiome. So you have that effect. And then the microbiome itself can become inflamed. Those inflammatory cells can enter the circulation. And as we've said, those inflammatory cells can enter the brain. So we think there are multiple ways in which the microbiome may be working in Alzheimer's disease. We can see that in the transgenic mice that are created to mimic Alzheimer's disease, that they do have a different microbiome. And it's very interesting. In the very early young mice, 3-4 months old, microbiome is normal. As the Alzheimer's disease matures in the brain, the microbiome changes quite dramatically. And there are drugs. There was a drug approved in China called GV971 or oligomanate that specifically targets the microbiome and is aimed at reducing the inflammation in the microbiome in order to reduce the amplified inflammation in the brain of the Alzheimer patient. So this is a really hot area. I think we're going to see more microbiome drugs coming along, and the preliminary steps have been taken in terms of microbiome therapeutics.
Okay, looks like we have time for one more question. Michael typed another one in. Are there promising behavioral or non-therapeutic treatments to manage early disease that you recommend?
Again, Michael, really great question. I haven't talked about lifestyle interventions. That's an intervention, but a non-pharmacologic intervention. It really looks good that if you take care of your brain, you can delay the onset of Alzheimer's disease, possibly prevent the Alzheimer's disease, or slow the Alzheimer's disease if it's present. We think, of course, exercise has good data on this point. Good nutrition with high antioxidant content has good data on this. Connectivity, living a life of purpose, controlling cardiovascular risk factors, good sleep, reduced stress, reduced depression. All of these things seem to come together in a lifestyle mandate that has the ability to reduce the risk of Alzheimer's disease. And it looks like about 30% of the total risk of Alzheimer's disease is attributable to lifestyle factors. So this is a non-trivial impact if we could get people to adopt a more brain healthy lifestyle. In terms of devices, there's a lot of work on stimulation, brain stimulation. Some of these are having interesting outcomes, and I think it's just too soon to know for sure whether there'll be a therapeutic effect and a sustained therapeutic effect. So there's a lot of work on non-drug interventions in Alzheimer's disease. There's also some cognitive therapy programs that are aimed at stress reduction and behavioral improvement. So there's a lot of work in the pipeline outside of drugs. Today, I've emphasized drugs because that's my interest in terms of clinical trials.
Okay, wonderful. I'd just like to thank everyone who joined us today on Dr. Cummings' webinar. And Dr. Cummings, thank you for your time, your insight, and your expertise. For the audience, if you have any additional questions or would like to connect directly with Dr. Cummings or the ClinLabs team, you can either reach out directly using the contact information that you see on this slide or contact us via LinkedIn, Facebook, or Instagram. Thank you for your time and have a wonderful rest of your day. Bye.
Great. Thank you, Shannon. Thank you,
Thank you
everyone.
so much.
Bye now.
Bye-bye.